Of course, before starting the lecture itself, I would like to warmly thank the Société des Neurosciences and its board for giving me this opportunity, the opportunity to share with you what was the core of my scientific thinking. I would like to thank again Patricia, but also Luc Buet, this very dynamic president of the society, and also the next one, Annie Andrieux, with whom I share some secret, the secret of hidden places in the Alps, she will recognize probably. And it's really an honor for me to give this lecture named from Alfred Fessard, the great French neuroscientist at the origin of modern neuroscience in our country, especially also because I worked for about 15 years in the institute that bears his name, the Neuroscience Institute Alfred Fessard in Gif-sur-Yvette. So now I would like to convince you that comparative neuroscience brings not only key questions to better understand how the brain is organized and generates actions and behavior, but it also brings, I think, compelling evidence and answers to many areas of neuroscience. As Patricia has already said, and I think as many of you, I have been fascinated by the tremendous diversity of nature, and this is especially true for the brain and the very large diversity of species that have a brain. Thus, the question was to know how to deal with and how to make sense of this diversity. The idea being that better understanding the organization and function of the brain will help us to decipher mechanisms, principles that eventually support and generate the behaviors of organisms in their milieu. So for this, we need to follow the path of the most prominent figure in evolutionary biology, Charles Darwin. And I cannot resist, to the pleasure to make this quotation. It's very famous. It's the end of the origin of species, but it's so beautifully written that it always amazed me. But this comparative aspect of neuroscience was just a curiosity for me for a long time until I became confronted to problems that forced me to enter into the field. And to do that, in fact, I followed a molecular path. And also, as Patricia has already said, this path was also being directed by dopamine systems. And here I will speak about three aspects of the dopamine systems in the light of evolution. The aspect of genes by speaking about the gene diversity, mostly of the dopamine receptors, and then of dopamine as a system of neurons, which makes dopamine. And the third point would be about the functions which are modulated and regulated by dopamine. So let's start by genes. In fact, genes were the reason why I got interested in evolutionary biology. At the time when I started in the 80s, the prominent idea was that gene evolution followed the principle of selfishness. This was introduced by Richard Dawkins in this very influential book, The Selfish Gene. And for me, it was really a shock. And it was a game changer, in fact, in science. Because according to this view, the major forces for organisms to evolve was that genes have to be transmitted, whatever the means for it. The most successful organisms are those which transmit the most efficiently their genes. So you see, there is no goal in evolution, no design, no intention. The successful, again, the most successful are just the one who transmit the gene the best. So I think it remains really true, but very soon I came to the idea that in fact this gene-centric view was missing also some crucial points. I think it probably missed something which is very important, which is, for example, the requirement of correct physical development of embryo, which really put a strong constraint on genes, and also how the environment shape organisms' traits, how this environment needs plasticity and impose a kind of variation in the genes. So genes, in fact, are maybe selfish, but not only. And maybe now you better understand the subtitle of this talk, which is selfish genes with obligatory constraint. Because I view genes, not really as these very beautiful green fish, which are free enough to jump out of the water and over the Chicago Bridge, but these genes are more closely resembling this poor goldfish, you know, which is jailed more or less in this aquarium, where he has to fulfill the constraint of functions. And I think it is these two aspects of the gene which were really fascinating, and I will come back to that very soon now. So first, speaking about genes. And to start with, I will speak about genes encoding dopamine receptors, I think as a good example of how genes evolve and how they can underlie functions. So when I started to study these receptors, I started studying them because as a neurologist, I was interested in better understanding the plasticity which occurred in the brain of patients after being treated by Parkinson's disease. And dopamine at that time was thought to act through two classes of receptor. This class of receptor were defined according to pharmacological principle, the specific actions of agonist and antagonist, different from the D1 and the D2 class. And also, by the way, they were activating intracellular pathways in the case of the D1 receptor, increasing the level of cyclic AMP. And in the case of the D2 receptor, mostly modulating ion channel, but also decreasing cyclic AMP. So it's mostly, at that time, there were these class of receptor were just entities, pharmacological entities without real biological not significant, sorry, but existence in terms of molecules. But when, with the advent of molecular techniques, there was a kind of explosion in the early 90s of cloning of the receptor of many, many families. And of course, the dopamine receptors did not escape to this rule. And here came two major, I would say, discoveries. The first one was to discover that, sorry, I don't see very well my screen. Okay, that the first major point is that in fact, these receptors, these receptor proteins were belonging to families, meaning they share similar structure and similar functions. In that case, the possibility to modulate the activation and the activity of heterotrimeric G proteins. And the second major discovery was in fact that where you had a single class of receptor, D1 or D2, in fact, each of these classes corresponded to several molecules, to several different genes, encoding either D1-like receptor or D2-like receptor. And we first, several groups were involved in the cloning of 3 different subtypes of D2 receptor and 2 different subtypes of D1 receptor in human. And these 2 classes of receptor corresponded to genes with no introns in the case of the D1 receptor, with several introns in the case of D2, and with very specific characteristic in terms of structure. So this molecular diversity was really a problem for me. Sorry. Because I wanted to try to understand what this molecular diversity was good for. And it's here when I met André Adout. André Adout is, beside being a really great person, was also one of the best specialists of molecular evolution in France. He was a professor in Orsay and then directing one of the most prominent research institutes in Gif-sur-Yvette. And I met André and I discussed with him about this problem of genetic diversity, because I said, I don't know why you need 2 classes and why there is such a molecular diversity inside each classes. And of course, André, a very wise person, told me, you know, in biology, when you start with a why, you will never get answer. In fact, there is no, as philosophers says, no proximal reasons. So you have better to try to understand the necessity for which these genes existing and what they are for in terms of adaptation and natural selections. And he advised me to change these why questions to 2 other questions: how this molecular diversity was generated. And we know now that it's by the mechanisms of gene duplication. And the second question is when. And for this, you need to study gene history. And he put me in the hand of Hervé Philippe, also a very talented scientist, who learned me how to use molecular phylogenies to reconstruct the history of these genes. And the principle of molecular phylogeny is pretty simple. You first align sequences, and then you count the difference between the sequence. And when you see in that case where you have only one difference between these two sequences, whereas you have three, it means that in fact these two sequences are more closely related, evolutionarily speaking, than the other one. And based on this parsimony principle, you can classify and reconstruct, based on sequence identity, which represent to some extent the time passing by, then the history of these receptors, of this gene. Sorry, in my case, yes, it was gene encoding receptors. And if you can then compare, the gene tree, you can reconstruct based on these molecular phylogeny with a species tree. And here you have a very simplified tree of vertebrates. And in fact, I'm sorry, Patricia, I will not speak about Amphioxus or Cilia. I will stick to vertebrate today. I cannot speak about everything. But here you have this simplified tree, and by comparing the tree of species with the one of genes, you can, for example, here highlight the fact that probably the genes were duplicated in the ancestor of ray fin fishes and the other vertebrates group. And thus, this genes duplication led to two different subtypes in some species, like mammals, with gene loss in other species, like amphibians here, for example. And it's exactly the methods we applied to reconstruct the molecular phylogeny of dopamine receptors. And to do that, we needed to have to clone at that time, and now to dig into the database of as much species as we can from the phylogeny of vertebrates. So here from the cyclostomes, so lamprey and hagfish, up to mammals and birds, snake, lizard. And by taking as much sequence as we can, by doing this in a proper way, we can reconstruct the history of genes. I just want to make another point to better follow me in these presentations. I would like to remind you that it's quite well established now that in fact, at the origin of vertebrates during evolution, 2 rounds of whole genome duplication occurred. So the duplicated of the ancestral vertebrate duplicate 2 times. The first one very close to the origin of vertebrates themselves, and then a second time, at a time which is a bit disputed, between the origin of jawless vertebrates, cyclostomes, and jawed vertebrates, the gnatostome that Patricia had problems to pronounce, which means the vertebrates with jaws. So these 2 rounds of duplications, and though the organisms with a double genome, more or less, with more genes, which probably was something which was very favorable to favor the diversity of their spread all over the world, and also the different physiology they have. And there is another round of whole genome duplications, which was occurring only in the ray fin fishes, and specifically in the Taylor's gene. So these fish have another round of whole genome duplication, meaning that most of the time these fish have more genes than the other group of vertebrates. So the first, I would say, results we got came as a surprise, but we went from surprise to surprise. So the first, I would say, prominent data was to realize that in fact the two class of dopamine receptors, D1 and D2, were not really related to each other. In fact, they are quite distantly related, since the D1 receptor is mostly related to other genes like beta adrenoreceptor or 5-HT4 receptor for serotonin, whereas the D2 receptor is much closer to alpha-2 adrenergic receptor or 5-HT2 serotonin receptors. So you see that their evolutionary origin is really different from the 2 subtypes, but still the 2 of them converge on the use of dopamine. So then we did, and here you have a list of people who collaborated to this endeavor, which took close to 8 years from beginning to the end, to clone the receptors or to look into the database to be able to reconstruct an history of the D1 receptor class, which is, I think, invertebrates now close to be completed. And in fact, in addition to the two first subtypes, which were discovered in human, D1 and D5, or in human and other mammals, we discovered two other subtypes, D6 and D7. I used a nomenclature which could be universally understood. This is the problem of nomenclature of receptors is also a big one, but here I will not address it. Just here you have just to remind that in fact, in addition to the two receptors originally cloned in human, we found two others, and they clearly occurred from specific gene duplications. And these four subtypes are present, for example, in birds, snake, lizard, turtles. They are also present in teleost fish, but they are not all present in amphibians. In amphibians lack the D7. The cartilaginous fish, the sharks and the rays, also lack the D7, and the mammals have only two of these receptors, D1 and D5. And here you see that in fish, you have not only 3 or 4 subtypes, but 7, because this is the mark of the extra whole genome duplication that happens in teleost fish. And the story is about the same for the D2 class. So first in human was identified D2, D3, and D4. And in fact, the 3 classes are indeed found in most group of vertebrates, from cartilaginous fish to mammals. But we discovered 2 other subtypes, D9 subtypes, which is absent in mammals, but present in the group of very tiny group of fleshy fins fish like coelacanth or lungfish. Also quite old originating species, which kept this D9 receptor. The fish also have this one. And again, you see that in fish, you have an extra round of genome duplication because, again, they have 7 subtypes of dopamine receptor. So you maybe in genetic terms, you see that fish are not simplified as compared to human. They are genetically clearly more complex. So this is the summary of the history of this duplication of these genes can be described as it is here. So in the ancestor of before the origin of vertebrates, so in tunicates, for example, or in cephalochordates like Amphioxus, the two classes of genes existing, the D1-like and the D2-like, and you can find them in the genome without any problem. So then a first round of duplication occurred close to the origin of vertebrates to duplicate the genes in the D1 class and the D2 class. The second round of genome duplications give rise to the 4 subtypes, or 5, because there is an extra one in teleost, you find in the modern organisms. So now we quite well understand this receptor history. Okay, so the other questions I was asking was, why are genes conserved? Why we still keep these genes in modern organisms? Because you may think that after duplication, these genes could, just disappear if they are not needed. So they may be maintained for 2 reasons. So either, because of selfishness and neutrality, they can be kept in the genome because they are there and they don't do anything really wrong or right, maybe, but they can stay. And this is possible. This happens from time to time. But also you can imagine that because they are born and because they brought new advantage to the animal, they have been kept for fulfilling this constraint. And it's what we have wanted to test a long time ago. And maybe before I show you the data, I would like maybe to really specify what I'm meaning by conservation, because I think it's not always easy to be precise in this manner. And here I really mean conservation, the presence of any feature in many different related species. It can be anything which is shared by or common to different species. And this can be applied to gene and protein. Gene and protein can be conserved, but also mechanisms can be conserved. let's say, regulatory pathways, for example, production of cyclic AMP, excitability of neurons, the biophysics of cells, the interaction they have, the forces building organisms. The morphological structure could be also conserved in anatomy. You may conserve the wings, legs, cortex, thalamus, and so on, or not. The functions can be conserved. So this is physiology. You can conserve flight, working memory, language, and I will come back to that later. And you can also conserve behaviors in ethology, social learning, hunting, for example. So this really corresponds to a Boolean logic of the, with the three main characteristics of or, not, and hand. So in the case of the or Boolean logic, it means that the characters are the ones which are shared by all the organisms together. They are found in this one or this one or this one. You have also the not logic. In that case, you will characterize the character which are present in this one, but not this one, but not this one. Or you have the one which are shared by everyone. It's the hand logic. So in that case, what I mean by conserved is the one which are shared by all the species considered in the study. I think it's very important to have that in mind to then follow what I will tell you. Then speaking about gene diversity and to try to answer what is good for, I have just to introduce you to the notions of subfunctionalization and neofunctionalization. So most of the time where genes are duplicated, they give birth to two identical genes. So when one of the two genes is, struck by a mutation which really make it deleterious, it doesn't matter because the other gene is there and this gene is disappearing as pseudogenes. But from time to time, the two genes take part of the functions of the ancestral gene, and the two genes take part of these functions. So in that case, because they share the original functions, they need to be maintained for the functions to be also maintained. But also, since you have two redundant genes appearing after gene duplications, one of the genes can, make innovations, and if this innovation is something which may be adaptive for the organisms, it will be kept. So it's how we can understand, the reason why genes are conserved. And in the case of receptors, in fact, we had to define which characteristics can be modified for conservation. And in fact, we can imagine 5 major characteristics. So first, the affinity for the ligand, for the natural ligands and efficacy of transductions. It can be the nature of the signaling pathways. For example, cyclic AMP versus, productions of calcium through IP3 and so on. It can be regulation of the receptor through desensitization and recycling. It can be the localization of the receptor in neurons, either presynaptic or postsynaptic or synaptic versus somatic and so on. And of course, it could be the regional distribution, let's say in the thalamus versus the hypothalamus and so on. So for all these reasons, in fact, you can have subfunctionalization or neofunctionalization. So we wanted to test this hypothesis, and this was a quite long work we did in collaboration with Chaim Niznik, and it was also a wonderful friend, and I spent more than 3 years of collaboration with him in Toronto, and I was not always in Toronto. I came just from time to time, but we collaborated to try to decipher, which kind of characteristic each type of dopamine receptors really can carry. And in fact, I just show you 2 of them. If you look at the black bars here, you can see that in 3 species, in a fish, eel, in an amphibian, Xenopus, and in human, in fact, this D5 receptor here has an intrinsic basal activity, which is double to 3 times more elevated than the one of the D1 receptor or of the D6 receptor. So the D5 receptors has clearly a higher basal activity. And when we looked at desensitization, you see that the D5 receptor, desensitize extremely fast and very efficiently, whereas the D1 receptor desensitized, but with a little delay. And the D6 receptor was not desensitizing sensitizing at all. So here, again, you see that we have clear differences in terms of functions and regulation for the 3 different subtypes of the D1 receptor. And this also, here you have the name of the people involved in this study. So we could then figure out why maybe these 3 different subtypes have been conserved after duplications. In fact, it's because they have different affinity for dopamine, with high affinity for the D5 and a lower activity for D1 and D6. The D5 have also a fast desensitization, so it make it a very, I would say, adapted receptor to be a neurohormonal receptor, what it is in fact. Where we have the D1 receptor with a lower affinity is clearly something which is more perisynaptic. There is no synaptic receptors in terms of dopamine receptor. They are always around the synapse, but not at the synapse itself. And the other major characteristic, I have no time to speak, is that in fact these subtypes are also located in different brain areas. For example, the D5 receptor is quite specifically present in some area of the hippocampus in many species, which is not the case of the D1 receptor, which is present in the striatum, for example. And the D5 is also present in some place in the striatum, but not at the same level. So you see, we clearly have, after gene duplications, the acquisition of specific new properties, which are either functional properties or differential regional expressions, which eventually constrain the conservations. And again, this conservation means adaptation. So I think there is a bright future for comparative molecular approaches in the neuroscience field still now. It's a very useful methods to better understand the origin and history of molecular regulatory pathways. It's also help for rationalize molecular classifications. It's absolutely mandatory when you want to, for example, do pharmacological approach in different species. You really need to know if they have the same kind of receptor or not. And more recently, I think these phylogenetic approach have been applied to structural biology to better understand, for example, the interface that this protein make with others in macromolecular complex by using artificial intelligence and taking advantage, of the large comparison of genes you can make to see which one is conserved and which one is not. So yeah, I think it's what I wanted to say about genes at that time, or at least about receptors. Now I would like to turn to another aspect of dopamine, which is the way it is acting as systems in plural. So dopamine is very well known, and I would like to make here a tribute to Jacques Lovinsky, who always has helped me during my career and very often also helped a lot for many different reasons. But coming back to dopamine, dopamine is, a neuromodulator which is regulating a large variety of functions, like, for example, olfactory, sensitivity, decision-making, learning and memory, chemosensitivity in the brainstem. And so a large diversity of functions are regulated by dopamine. And this is what I would like to briefly address with you. First, we have to define what is dopamine neurons. And in fact, the dopamine neurons is defined by a chemical phenotype. And this chemical phenotype starts from the transformation of an aromatic amino acid, L-tyrosine, into L-dopa by the key enzyme of the system, tyrosine hydroxylase. And then L-dopa is transformed into dopamine by a second enzyme, aromatic amino acid decarboxylase. Then it is packed up into the vesicles through a vesicular monoamine transporter and then released outside of the cell and acting on target cells. And the system is stopped by uptaking back dopamine into the cells through a specific dopamine transporter, and then degraded by 2 enzymes, either monoamine oxidase or catechol-methyltransferase. So in fact, it is the presence of these molecules, tyrosine hydroxylase, aromatic amino acid decarboxylase, VMAT, and DAT, which really specify the phenotype of neurons, making them dopaminergic neurons. So it's what we have used to try to study, in fact, the way dopamine systems are organized in different species, mostly fish, birds, and amphibians all over the years, and compare them to mammals. And here I just would like to emphasize one very important point. In fact, if in mammals or in birds or fish, for example, where quite a lot is known, each neuronal group making dopamine, which have been classified a long time ago by name from A1 to A17, it's the number of it's the name of it's the nomenclature, sorry, classical nomenclature of the dopaminergic neuronal groups in the brain, and each, in fact, of these neuronal group is during development specified by different genetic pathways. So there is no genetic master gene, so no genetic logic in the way dopamine neurons are made. They are made by many different regulatory paths. Each system is more or less independent, and probably each of these systems has been recruited independently during the evolution of vertebrates. So it means that, as you will see, there are significant variability among species. And also, we are really to be fully aware that there is no one dopamine system, but many dopamine systems. So I just would like to, in fact, speak about two of them. The first of them, which is something quite astonishing, in my opinion, you will tell me, is the one of the paraventricular organ, which is present in many species of vertebrates, but not in placental mammals. This region doesn't exist in placental mammals. First, I have also to tell you the fact that, in fact, tyrosine hydroxylase in vertebrates is an enzyme which is also encoded by two genes. In fact, for a long time, only one tyrosine hydroxylase was known in human and mammals. But almost 10 years ago, we discovered that, in fact, there was a second tyrosine hydroxylase in the genome of the chicken, for example, of the xenopus or of fish. And in addition, as you can see here, this Th2 gene, in fact, is located between neighboring genes like IGF-1 or PAH or IKH2-like1. And this synteny is conserved in xenopus, is conserved in chicken, is also conserved in human, except you see here you have no Th2. So it's pretty clear that, in fact, these genes, the Th1 and Th2, originated again from genome duplications which took place on this ancestral chromosome, giving rise to the two copies of IGF-1 and IGF-2, for example, or Th2 and Th1. And this duplicated genes has been lost, in fact, in not really in mammals per se, but in placental mammals, meaning that placentation was probably something which has something to do with the disappearance of these genes. This is a very interesting story, but I will not tell it you here again. But here the major point is that you have to be aware that in fish, for example, you have, or in birds, you have two TH and not only one. And this helps us to solve a long-standing controversy in the field of comparative dopamine systems. In fact, it was known for a long time that in brains of birds, for example, but also true in amphibians or fish, you have here a region lining the ventricule, which is called the periventricular organ, which is making dopamine. This is an immunocytochemistry against dopamine itself. But curiously, no tyrosine hydroxylase, no enzyme making dopamine was found there. So for a long time, these cells were thought to be dopamine accumulating cells because they were not thought to be able to synthesize dopamine. In fact, Kei Yamamoto in the lab was curious to look at that. And in fact, she discovered that, in fact, Th2, the second enzyme, is in fact present in these cells, and that very likely they are able to make dopamine, in fact, and they were not accumulating cells only, but making cells. And this is also true in the zebrafish. You see here the immunoreactivity for dopamine in green, and you see that the immunoreactivity for tyrosine hydroxylase itself is not there in these green cells. It's just in a few big cells outside. But if you make an in situ hybridization for Th2, you see that, in fact, Th2 is indeed expressed in these green cells. So you can see it maybe in a more precise fashion if you just look at this population here of cells, you see that they have dopamine clearly, but they have no tyrosine hydroxylase at all. And Kei Yamamoto and people in the lab were able to show that, in fact, these cells, in fact, indeed express Th2, but also VMAT and the aromatic, the VMAT, it's the vesicular monoamine transporter, and also the aromatic amino acid decarboxylase, meaning that these neurons are really bona fide neurons and that they really was making dopamine and not accumulating them as it was thought. So the paraventricular organ was already something a bit, I would say, weird as compared to what you maybe knowing in mammals. But we also a few years ago discovered another very large unexpected feature of these PVO cells is that the fact not only these cells have dopamine, but they have also serotonin. And they co-express, in fact, in about 20 to 30% of the cells, serotonin and dopamine. And it was something which really was not known before, and something which was even not thought to be able to exist. So in fact, when you think about it, and when you think about the way these phenotypes are defined, it's not so surprising. The reason is this one. If you compare the phenotype of a dopamine neurons, the one I explained you before, and the one of the serotonin neurons, the major difference between them is, in fact, the nature of the aromatic amino acid at the start of the synthesizing pathway, L-tryptophan instead of L-tyrosine, and the fact that the enzyme is thus a tryptophan hydroxylase and not a tyrosine hydroxylase. But these two enzymes are really closely related in terms of both structure and function. But then, as soon as this enzyme has acted, the rest of the enzyme, aromatic amino acid decarboxylase, is the same in dopamine neurons and in serotonin neurons, and also the vesicular monoamine transporter, which is making dopamine to accumulate in vesicles or serotonin to accumulate in vesicles, is the same in the two neurons. And then only the membrane transporter is also different, even if, again, they share a lot in terms of both structure and function. So in the case that I just showed you, we were wondering whether these cells were also expressing TPH. And in fact, it was the case. And so we already knew they had Th2, but we were also able to show that they had the TPH enzyme, making them able to synthesize serotonin in addition to dopamine. So these cells have a double phenotype, dopamine and serotonin. So you will ask me what they are doing. We don't know very much. It's not so easy to study the physiology of these cells. The only clue we have came from studies in birds in which they may be involved in seasonal rhythm. The fact that organisms have to adapt, in particular their reproductions, to seasons. And there are some evidence that these cells could play a role in this, but there is no other evidence in other species. So the conclusion of this part is that, in fact, this modularity of the system, the fact that you have only 4 molecular components at the origin of the phenotype of dopamine favor probably the independent recruitments of dopamine phenotype in different brain regions and different neuron types. I have shown you that you can have the coexistence of several monoaminergic phenotype in some brain areas, like the paraventricular organ, and this modularity and versatility, I would say, favor contingency and species adaptations to different way of life. And the consequence of that is that, in fact, there are quite a strong variability in dopamine systems among different vertebrates. So again, you have to be, I would say, very cautious when you would like to directly compare, for example, zebrafish to mouse or human, or if you want to directly compare birds to other group of species. But at the same time, it's clear that dopamines and monoamine in general are really phenotypes which are very well suited for neuromodulation. So now the third questions I would like to address with you is the one of functions, and this is a pretty tough one. So I will try to be as simple as I can, and I will take a single example, the one of substantia nigra, and ventricular tegmental area, which in many species are mandatory to control functions like motor programming and working memory through afferents from the substantia nigra ventral tegmental area, either on the basal ganglia or in the cerebral cortex. And if you look in different groups of species in vertebrates, from turtle, lizard, crocodiles, amphibians, mammals, you see that this substantia nigra ventral tegmental area is pretty well conserved. In fact, these dopamine cells are located in the mesencephalon of the species. And in all these cases, they also project on the striatum and the cerebral cortex to a different extent. It is very well known that these dopamine systems originating from substantia nigra ventral tegmental area are really mandatory for the existence of working memory by projection they make in primates on the prefrontal cortex, and by the projection they make in the very complex prefrontal cortex of primates and the more simple prefrontal cortex of rats, for example. These animals have a 6-layered neocortex. And in the case of birds, it's a bit different because birds have no cortical structure in a true sense. They have instead a pallium made of different nuclei. I will not enter into this debate here, but it's well known and well established now. And the astonishing feature is that, in fact, in birds, it has been shown by Gunterkun and others that, in fact, the cells originating from the substantia nigra VTA and which are really mandatory for working memory to occur are not projecting to frontal regions, but instead of posterior regions of the pallium, the nidopallium caudolaterale. And in fact, the group of Gunterkun and others have shown that, in fact, this region of the pallium of the bird is playing the same role than the prefrontal cortex in mammals. And here you have a very nice example of functions which is maintained, but through different anatomical connectivity, through different anatomical organization of the system, even if it's still dopamine doing the job. So in the group, Kei Yamamoto and a very talented PhD student, Solal Bloch, with the help of Cynthia Frock, wanted to see whether also motor learning and mostly working memory was existing in the zebrafish. And by using a test of delayed matching to sample in the zebrafish, where, after presentation of the cue, the fish has to wait for a couple of seconds before choosing either the green of the red compartments. So it's really the same kind of working memory test you can do for example, in mouse or in birds. And Kay and Solal were able to show that, in fact, the fish can learn this quite well, and they really have a very decent working memory. Again, very, very comparable to the one of a mouse, for example. The only problem we got that, in fact, in fish, you have no dopamine cells in the mesencephalon. And in fact, it's even worse than this, because for some time, we thought that maybe a group of dopamine neurons present in regions called the posterior tuberculum of the fish could play the role of substantia nigra, because with Mario Voliman and her student, Elke Rink, we showed that some of these cells can project to the striatum in fish. But in fact, it was more recently shown by the group of Driver that this is clearly not true. First, these cells are not differentiated at all by the same mechanism, the one of substantia nigra, and then they are mostly projecting over the spinal cord. So there is clearly no homologue of substantia nigra in Tiller's fish. So the question remains to understand how working memory could be occurring and maintained in zebrafish. So I think this is very important that now we can have in mind that several conserved functions can depend on different anatomical organizations and networks in different groups of species. But at the same time, I think it's very interesting because it stressed the fact that constraint functions can oblige the system to converge on through different solutions on similar principle of organizations to convey the functions. So I think that these days, comparative neuroscience are really very important to elucidate the principle of functional organization that underlie functions, and that concept functions can rely on different anatomical and physical organization. And we badly need better neurophysiology in as many organisms as we can to try to really pinpoint these questions. And to finish, I would like to emphasize the fact that I think this elucidation of principle of functional organization and underlying functions like language, memory, motor control, sensory representation is really something which is of utmost importance for current neuroscience. And I think that large flagship program like Human Brain Project or eBrains now should tackle these questions in addition to only considering human, because you can then better understand the principle which really target the functions and not a specific organization that you can find in human, even it's of course of extreme interest. But I think the power in terms of computing, in terms of applications in sensory, in science of information is really very, very, very important. So I would like to now acknowledge my team, my dream team, I would say, and especially Michael Demarque, who really helped me over all these years to set up the group. Of course, Kei Yamamoto, a very talented, very productive, a very active scientist. I showed many of her discoveries in this talk, and also Ingrid Collin, our engineer, also extremely active and talented. Jean-Michel Hermel, again, the other engineers. Cynthia Frock, we help us with fish behaviors. The students, the current student, Pierre Lestienne, and the previous one, like Solal, Romain. All the other people, my long-standing colleague Catherine Pascalini, postdocs like Anna Havir, and many others, all the collaborators at TEFOR, in GIF, Pierre and Arnim, many collaboration in France, the collaboration I've established all over the years in different countries. I will not read all of them, the fundings, of course. And then maybe, since it's a Fessard lecture, maybe you will allow me to make some more personal, maybe presentation and thought. As Patricia also has said, I spend quite a lot of my time in organizing things in the Paris area, and mostly in the Paris South area. And I'm quite happy that now we can be, the following of a long suite of people from Alfred Fessard to now François Rouillet, who is the head of the lab. We will move very soon now in these new buildings that I have dreamed of for 10 years now, and we really push for this just in front of Neurospin, and I think it will be very exciting to have both of these two institutes in front of each other. And also now to be even more personal, I would like to use, in fact, the words of a great poet, a great Scottish poet, Kenneth White, who I think capture the whole nature of evolutionary biology by saying that for the question is always how, out of all chances and changes, to select the features of real significance so as to make the welter a world that will last. And how to order the signs and the symbol, it could be the genes in that case, so they will continue to form new patterns, developing into new harmonic whole. So keep life alive in complexity and complicity with all of beings. And in fact, when you are a scientist, it's a lot of work. It's very often discouraging, and it's an endless quest. And from time to time, I like to see this nature as a whole. And I think we, from time to time, can allow us to say that this is only poetry. And I think I was very, I would say, moved by the fact that recently I read a book of a great French poet, but also physician and surgeon, Laurent Gaspard, in fact, Patricia's father, and with the same idea. He said, and I will read it in French, he wrote in French, and I think it's so beautifully written that I cannot do something else than read it in French. And it's again about functions. It's astonishing, you know. Une fonction, du moins la biologie nous le montre ainsi, éclaire un fonctionnement. Nos linguistes se sont longuement penchés sur les fonctions de la langue, sur celles de la poésie. Planche admirable d'anatomie, développement ingénieux de cycles de chimie, de mécanismes de physiologie, on peut s'en tenir là. Qu'est-ce qu'en effet a tant de rouages sans défaut qu'une couleur qui hésite, qu'un mot qui manque, quand s'attarde au soir la lumière sur les eaux. And to finish with, it's moving indeed. I would like to say that during all these years, I've met many people, many friends. Many of them are still friends. I met a lot of good colleagues. We spoke hours speaking, making the world better, trying to make the world better, sharing food, sharing wine, discussing, you know, many things in science, trying to make all what we have done advancing in the right directions. And in fact, I always remember what another great people I met told me and told to students, used to told to students. Nicole Le Douarin used to say, when you are able to do research, you are really highly privileged. And I think this is more true than ever. Thank you very much.
Merci Philippe. Un grand bravo. Je pense qu'on peut t'applaudir. Je disais en introduction, vous connaissez Philippe, son côté académique, mais voilà, vous avez l'homme, et je trouve que là, la lecture Alfred Fessard retrouve toute sa force.
Merci Patricia. Merci Annie.
Alors, il est de tradition qu'il n'y ait pas de questions à la lecture Alfred Fessard. Il est 15h03, donc je voudrais remercier tous les participants. On était à près de 260 participants. Je vous demande de ne pas nous quitter, puisque nous avons, après cette lecture Alfred Fessard, l'Assemblée générale de la Société. Mais encore une fois, un grand merci à Philippe pour ce partage scientifique, mais aussi humain. C'est l'image que tu donnes, hein, Philippe, quand on te connaît bien. C'est vraiment ce partage et cette expérience humaine qui va au-delà des neurosciences. Donc, un grand merci. Je pense que tout le bureau et Annie s'associent à moi aussi pour vraiment te remercier pour cette lecture.
J'espère qu'on pourra fêter ça ensemble autour d'un verre quand même un jour pas trop lointain, parce que c'est vrai que c'est assez frustrant de faire ces présentations à distance comme ça. Mais il faut se plier aux lois du temps.
Malheureusement, bon, on sera peut-être tous à Paris pour la FENS en 2022, donc ce sera peut-être l'occasion. J'ai vu qu'Alain était aussi là, donc